Welcome everyone to the Human Cell Atlas Developmental Seminar Series. Today we will be talking about spatial cell mapping with an exciting list of speakers and panelists. So we'll move to the next slide. So in order to get greater audience participation, we are trying out slido.com /HCA 2020, which is the link you should go to interact during the seminar. You can post questions and so on and so forth. And you can also do that by scanning the QR code on your phone, and the QR code is here. For questions, please submit your question at any time. Again, at the if you access the Q&A tab highlighted in red here at the website, and questions with the most votes will be prioritized, and we'll be asking them after the two speakers. In addition to the Q&A, we've also got an resource area, which is under ideas, circled here in red. So if you do want to contribute any resources related to spatial genomics, just put the resource or the link to it there. That would be fantastic. We will also be undertaking a quick poll, and we want you to participate. We want to hear from you. Two questions. One is, what would you like to get out of this seminar? And that's at the poll link of Slido. And if you can start answering your questions now. Oh, my name is Ms. Hanifa. I'm your, that's one of the questions. So this is go on. So please do submit your answers to the poll. So it looks like new ideas is the thing that most of you have answered, and we'll be presenting the outcome of the active poll perhaps at the end of the introduction. So I'll go to the next slide. Oh, you've answered that question as well. How did you hear about this seminar? And basically the vast majority of you have answered that direct email is the best way to actually get in touch with you. I'm quite surprised by the response for Twitter and Facebook. Yep, that's brilliant. So keep going with your answers, because I think there are more than 500 of you here today, and there's 129 response. So that's brilliant. We'll move to the next slide and give you the answers to the poll later on. My name is Ms. Hanifa. I'll be your session host, and assisting me are Bruce Arano and Gary Bader. We have two guest speakers, Mats Nilsson and Ali Ertug, and we also have a fantastic panelist who will be assisting our speakers, and that's Anne Shadatal, Joachim Lundeberg, and Xia Wang. So we now move on to the seminar, and first on our speakers list is Mats Nilsson. Mats is now the site director of the SciLife Lab in Stockholm, and looking forward to your seminar, Mats, and thank you very much for agreeing to participate.
Thank you very much, and thanks for inviting me. So I'm Mats Nilsson. I'm at the SciLife Lab in Stockholm, and I'm a professor of biochemistry at Stockholm University. I'm going to talk about our approach to draw spatial cell atlases of developing organs. And I'll start with the disclosure. I'm co-founder of Cartana, which is a company commercializing in situ sequencing products. But in this, the data that has been generated in this presentation has not been using any of Cartana products. So, development in time and space. As you all know, development is driven by cell line intrinsic epigenetic programs, but also by external molecular cues such as concentrations of morphogens and direct interactions with surrounding cells. Birth of new cell types and developmental lineages can be great sensitivity recorded and timed using single-cell RNA sequencing. This method also very nicely characterizes cell intrinsic factors. However, to understand the patterning of tissues and to directly observe the spatial determinants of the cell differentiation, it's spatial and molecular data needs recording. So I'll show you our approach to this and some illustrations of what can be done with spatial molecular profiling. So I'll first give a little bit of a background to the Swedish Human Developmental Cell Atlas Consortium. I'll talk about the method as such, the targeted in situ sequencing we have developed over the years. And then I'm going to show some very recent data that we have generated in collaboration with Sten Linnarsson on mapping developmental organizers in mouse developing brain. That's out on bioRxiv since 2 weeks. And spatial atlases of molecularly defined cell types I will describe, and an application of this approach to make a spatial atlas of human developmental heart, a project we've been doing together with Joachim Lundberg. So the Human Developmental Cell Atlas Consortium in Sweden is focusing on brain, heart, lung, and actually we're also go on to provide some sort of atlas of a whole embryo as well. And we focus on the first trimester of development. And the participating principal investigators in this project are in some sort of logical order here. Erik Sundström at Karolinska, who is heading the biobank and the tissue retrieval for the project. And we have Christos Samakovlis here, who is the lung developmental specialist in the project. Sten Linnarsson, who is providing single-cell RNA sequencing data, and his lab has today processed about 2 million cells from post-conceptional weeks 5 to 13. Joachim Lundberg is providing transcriptome-wide spatial expression profiling using the spatial transcriptomics method. And then towards the end, we have targeted methods to do more detailed characterization of tissues. That's Emma Lundberg, who will map or is mapping sets of proteins onto the tissues, and my lab that is using targeted in situ sequencing to provide spatial cell maps of using targeted expression profiling, RNA expression profiling method. The method we're using, which we call targeted in situ sequencing, is a probe RNA expression method that works in cells, in fixed cells, in tissues. And we use probe molecules targeting sets of genes that become circularized when they interact with the correct target sequence. And the probes are barcoded to be able to identify the target genes. And after circularization, these probe molecules become amplified massively. So we get about 1,000 copies of the barcode sequences introduced into these massive DNA molecules that are produced during the Raman circle amplification reaction. These barcodes we read out by different combinatorial decoding approaches using 4 or 5 dyes, and then sequential labeling and imaging and washing and so on. And in, say, 4 cycles of stain, we can distinguish 256 different combinations of dyes, 5,000. And typically we do maybe 6 or 7 cycles of staining and imaging to get robust differentiation of all the barcodes we want to distinguish in cells. So first to the mouse brain mapping effort, and this is a massive work from Sten Linnarsson's lab to do comprehensive single-cell RNA sequencing of the developing mouse brain. So through different ages and different parts of the brains, and see how cells are born and populate this atlas of 1,000 different states of cells in the the mouse developing brain. So our part of it, yeah, and this was just recently published on bioRxiv, and you can also browse this data on SDN's mousebrain.org. So our part in this massive effort was to validate a prediction that was based coming out of the single-cell RNA sequencing about genes that seem to be specifically enriched and expressed in these organizers in the mouse brain. It's these regions, specific parts of the brain where a lot of the important development is happening. And they had identified 119 ligands, receptors, and transcription factors that seem to be important, expressed in these organizers. So we designed probes for the 119 genes and applied in situ on several series of sections of a whole embryo, and could confirm the predictions, and I could see that the genes fall very nicely into the different organizer zones. And one could even see with another set of genes that these organizers could be structured, so there are different subpopulations of cells that occupy subsets of or parts of these organizer regions. So all in all, by doing an unsupervised clustering of the spatial data, 40 distinct regions or environments could be defined that use different combination of these 119 genes. So each providing a unique environment for cell development. The other approach I'm going to talk about then is this probabilistic approach to draw spatial maps of molecularly defined cell types. And the whole objective of that approach is to be able to tell where cell clusters from single-cell RNA sequencing experiments are located in tissue sections. So to basically place all these clusters into space in the tissue. And it's a quite straightforward approach. We define cluster-specific markers, sets of cluster-specific markers to be able to identify in combination, every cluster of single cells. Design the probes for these genes and run the in situ sequencing experiment to generate molecular maps of the targeted genes. And then assign all genes to individual cells in the images, and then compare the molecular composition of every cell in the tissue with the average expression profile of the single-cell RNA sequencer clusters. Then we plot that as a probability. So this cell has a very high probability to belong to this particular, in this case, neuron in mouse brain. We validated this and developed it and validated it in mouse brain. And here is we focused on CA1 of hippocampus of mouse brain, and there wanted to draw a map of all the interneurons using a panel of 99 genes. And this is the map where one can see all the neurons in one CA1 region of hippocampus, and the corresponding interneurons that we have mapped here. What one can see here is that you don't really need very much information to do this. So it's about 30 gene expression observations per cell. You can still, with that limited data set, you can still, with very high confidence, map or assign a cell identity based on the single-cell RNA sequence, because you have made very specific selection of the marker genes that are expressed, and all genes are not expected to express in every cell. So this set of genes also did a decent work in mapping the neurons of isocortex as well, both the excitatory and the inhibitory. And then we took this approach to map a set of human developmental heart tissues. So this is the project we did in collaboration with Joachim Lundeberg, and so what he had generated was single-cell RNA sequencing data with a modest number of cells from post-conception week 6.5 heart, and also spatial transcriptomic data, so the pre-Vesium version of it, of 3 time points, so 4.5, 9, and 6.5, and generated clusters of spatially differentiated gene expression patterns. We draw marker genes from both these data sets and designed probe in situ sequencing probes for 69 marker genes, and then draw a probabilistic cell map of 15 single-cell RNA sequencing defined cell clusters. And this is what you see here. And in this illustration, you have we mapped almost 21,000 cells. And that was about 80% of the nuclei we observed in that tissue that passed the quality threshold for giving it a cell identity. And we also did the in situ experiment on the 4.5 and the 9 post-conception week heart as well. And one could also look at, try to identify corresponding regions in the hearts and see how they sort of develop over time. Certain cell populations will change their expression program, basically. All this is available on this portal, the HCA Sweden, scilaflab.se. The last thing is sort of a little bit more forward-looking, and this is also links to a little bit of a transition over to Ali's presentation. And I'm presenting here the Yugodeca project, which is led by Alain Charlatan, where we want to be able to provide deep molecular information to whole embryo or embryos that have been imaged with whole embryo imaging technology. Basically, take sections of imaged embryos, fill them with molecular information, and draw cellular maps across several planes of the developing organs, sort of to try to link the molecular data to the overall 3D structure of the complete embryo. And yeah, we're really a technology developing laboratory, and so we are improving the chemistries behind our methods, and so we are upgrading now the decoding of strategy that we're using now to get even cleaner and stronger signals, and we can also decode more crowded data. And soon we will have put on the bioarchive an upgrade of the basic chemistry in the RNA targeting. Those are the acknowledgement and my fantastic group, and thank you for your attention.
So thank you very much, Matt. We will have our Q&A with the panel after both talks. So we will move on to Ali's talk now. Ali is an inventor and an artist, as well as the director of Helmholtz Institute on Tissue Engineering and Regenerative Medicine. We had a slide to introduce Ali, actually, which I was very keen for us to put it up because it shows all of the incredible achievements his lab has made with regards to tissue clearing. And so he's been the director of ITEM in Munich since July 2019. So this is some of the things that he's getting up to in his lab, and look forward to his talk. Thank you, Ali.
Hi, everyone. It is great pleasure to present our recent research here to share the details with you. In my lab, we are very interested in combining different technologies from biosciences to AI and engineering to accelerate biomedical discoveries. And here I would like to focus on our biomedical imaging technologies that are merged in the last years with AI to accelerate human organ mapping efforts. Well, as you know, we are an organism of interconnected systems from head to toe. We are wired with nerves and vessels and lymph nodes and other systems. It's clear that a disease affecting one part of the body will affect the rest. However, in the lab, we rarely study diseases at whole body scale because simply we don't have tools to do so. If you just think about imaging tools, on the one side, we have MRI, fMRI, DTI-like imaging modalities, where we can see gross anatomy over time, but we don't have cellular information. If we want to study cells and molecules, we can do serial histology, serial EM, but the problem there, we cannot scale this up to a whole organ or a whole organism. So the technologies that we have been developing actually combines these two. We have tissue clearing technologies where we can now see gross anatomy at subcellular details in a fast and scalable way. So what is tissue clearing? As you know, standard histologies rely on tissue sections and labeling and looking at the fragments. This is most of the time works very well, but if you want to study complicated organization of 3D structures in organs, that is not sufficient. For example, looking at fragments of vessels and nerves. To overcome this problem, we and others have been developing so-called optical tissue clearing technologies, where we can convert opaque biological tissue into transparent structure. You can think this as converting milk into water. We can see through and now scan through to obtain 3D information of intact biological specimens. So when I started my lab, we started focusing on actually on whole mouse bodies, the most commonly used animal model. And in 2016, we developed UDisco technology where we could make whole mouse bodies transparent, but also we could image at that time for the first time with laser scanning microscope the mouse bodies. 3 years later, we developed the UDisco technology, which is now enhancing the signal that is appearing in the mouse bodies about 100 times now. You can see the nerves on the right-hand side is much more visible compared to green picture where was hidden behind autofluorescence and background signal. So UDisco actually truly renders the whole mouse bodies transparent. You can see it really like glass. After making them transparent, we put them onto light sheet fluorescent scanning light sheet microscopes and then scan them end to end with the lasers and see cellular details. And of course, doing so, we generate several thousands of images of whole mouse to obtain all the cellular information. And then we use machine learning to analyze this large data. We have been focusing on deep learning in the last 3-4 years because it is really unbiased and scalable, and we can do it very quickly. For example, Mike and Johannes, they have developed a technology to label whole mouse brain vasculature down to capillary level and then analyze using deep learning and register it to Allen Brain Atlas and obtain all information of vasculature in different small brain regions. We have been also very interested in applying these technologies in cancer metastasis. As you know, majority of the cancer patients, they die from metastasis of the tumor, and finding out these tiny tumors throughout the body is very important, and to make sure they are treated by drug is very important. But the technologies that are at preclinical research is the same at the clinical research. From bioluminescence to MRI, they will show you all similar view. To be able to see something, we will need to have thousands of cells together. So we are nowhere close to seeing tiny few cancer cells, maybe representing cancer stem cells and forming this large metastasis. And we thought we could overcome this problem, and Oliver, together with Chen Chen and Arnaldo, they developed this technology, so-called DeepMAC, where we put tumor-seeded human mammary carcinoma to mammary fat pad. 10 weeks later, it is this metastasis all around. Then we render the whole mouse body transparent and use deep learning and identify cancer metastasis, shown here represented in red at single cell level. In addition to that, we could also follow a targeting therapy here, use an antibody, 6A10 antibody, again at the cellular level. It looks like this. So DeepMAC, together transparency and deep learning, we can see tumor metastasis magenta and targeting efficacy of antibody here for this tumor that has been developed by our collaborator. So to give you a comparative image, so here is standard imaging of tumor using bioluminescence. You see for tumor signal in the rectangle where there is the lung, there is nothing. But after application of our technology, transparency plus deep learning, we could see actually there are hundreds of tiny micrometastases in this mouse body. Let's have a look at this in 3D. You see now the major metastasis close to the leg. But then if we go through, fly through in the mouse body, you see hundreds of tiny micrometastases distributed all around the body, in muscles, in the bones, and in the lungs. And you can really see the tiny metastases, small down to cellular level. In addition to that, we can see targeting efficacy of antibodies. Now, if a tumor is targeted, you see them now in white, and those that are not targeted, they are in magenta. You see this is an antibody-based therapeutic. Even then, we were surprised to see about 20 to 30% of the micrometastases, they were not targeted by the therapeutic antibody. That was actually quite surprising, because now we have to probably stop and think carefully before pushing such antibody-based drugs to the clinic, because we can see they are not ready, maybe missing a small micrometastasis. So those are some of the few examples on mouse bodies. How about human tissue? The same protocols. When we started applying on human tissue, it didn't work well. There was a problem, and Sean in the lab, a chemist, solved this problem. So what was the problem? The standard detergents like STS Triton used in tissue clearing and labeling, they didn't work well. They couldn't permeabilize this tough, stiff human tissue because of their large micellar size. So we then screen and identify a new detergent called CHOPS, which has this tiny micellar size and could go through stiff human tissue and permeabilize, make the holes. Then we could label and image the human organs. For example, you can see here, whole human eye is rendered transparent and then scanned with light sheet microscopy, and we can see the dye labeling plus autofluorescence that shows us the details of the iris and the retina and the nerves and immune cells behind that. So in addition to using dyes, we could also do actually large tissue antibody labeling. I already mentioned CHOPS helps us to do permeabilization, but also we identified two additional chemicals, acetic acid and guanide, HCl, that help us to loosen extracellular matrix, therefore allows doing antibody labeling on human tissue more than a centimeter thick. For example, here is a human brain cortex tissue labeled IBA1 in green. You can see the white matter, microglia, gray matter less, and again white matter, a lot of microglia. And there's millions of microglia. Not only their cell bodies, we can see also their morphological details. So what we can do also nice is that after doing this large scale imaging, if we identify some pathology, some small region, we can take it out and rehydrate this tissue and perform H&E staining, needle staining, or even regular antibody staining we could perform very well. So that is actually going from unbiased whole imaging to molecular analysis of small identified regions. Of course, imaging large human tissues like this, actually, we are looking at billions of cells to analyze that. Again, we are been developing deep learning algorithms here developed by Rami to analyze really hundreds of millions of cells within a very short time, within a few hours in the brain cortex. So we could label, clear, and image whole human organs. So what can we do with that? So we have a few applications. We want to, of course, map human organs. first to understand the developmental processes as how the biology works in health and what goes wrong in disease case. So we have a whole view of the organ and can study in a different perspective. For example, here we have a kidney from 92 years old person, post-mortem donation. We can make this kidney transparent again using channel technology, and then use the laser scanning light chain microscopes and look at the details. You can see here tiny capillaries in green, and we will see in magenta the glomeruli. You see on the left-hand side there are a lot of these capillaries, very as expected, but on the right-hand side there is this gap. We were surprised to see that. So these tiny capillaries were not there. When we look at in magenta channel, the glomeruli, we also did not see them in this empty region. There was almost no glomeruli, while on the left-hand side, again, there was a lot of one. So basically, consulting with kidney experts, they were also surprised to see something like this, simply telling us that looking at the whole organs, high resolution, gives us different views in their biology and if there is disease. We are very interested in, as a second point, mapping human brain. Well, it's actually clearly one of the flagship projects of all human sciences, and billions invested in the EU and in the US. Since more than 10 years of human brain project, there is no map of a human brain at the cell level. It's clear that a major problem, we don't have technologies yet to get access to cellular information in the human brain. But now, after making the human brain fully transparent using channel technology, at least we have now an access to scan the cellular information with the laser scanning microscopes. So at the moment, we don't have such microscopes. No one had made such a large microscope. We are also working on to develop a microscope that we can place this large organ and image. And finally, we are very interested in using these human maps, cellular maps, to do 3D bioprinting or replicating them using 3D bioprinting. So what is the connection? As you know, 3D printers are 3D replicating machines. If you have a 3D map of something, they will generate it. And of course, if you want to replicate a human organ in 3D, what we would need a 3D map of a human organ, at least at the cellular level. And when we generate now a cellular map of human organs, like the kidney, mapping of the tiny capillaries, glomeruli, and additional cell types, whatever we could profile, we will get mathematical formulation of a kidney, and then we can hopefully in the near future start generating from small scale to large scale human organs. And that's all from my side. I'm very happy, of course, the funding agencies, and very happy to work with a multinational, multidisciplinary team from engineers to physicists, chemists, and we are 16 countries in about 20 people. So I'm happy to take your questions.
Thank you very much, Adi, for this exciting presentation. We will now move to the Q&A session with our speakers and our panelists. For those of you who are attending this session, please submit your questions, which you can see the kind of the ranking of the questions by popularity, and so we will start with question that has the highest number of votes. And at the moment, the first question is directed specifically to Mats, and it says, how does one model spatial cell interaction, and how does one model spatial cell interaction, and how is it different from non-spatial? What are the additional challenges?
Yeah. Well, that was a hard one, I think. Could you try to rephrase that question or read it again?
Yeah, so basically it's saying, how do you actually model spatial cell-cell interaction, and how is it different from non-spatial? So I do think it's kind of quite a difficult question for me to even understand it, but I guess it's sort of like wondering whether, yeah, that's what it says. What are the additional challenges? And there are 9 people who like these questions. Yeah. This particular question, I mean. maybe we can think about it. And the other top popular
yes.
question from Trevor McKee, there are 14 votes for this, and this is for the panelists, for all of you. Can we define fundamental limits for spatial genomics or proteomics on tissue sections? That's the first part of the question. The second part being, as we go from whole tissue deep sequencing to tiled down to single cell or diffraction limited spots, the amount of material available drops. So how abundant does a transcript or protein need to be at the single cell level to detect it? Any offers from our panel members? I can't see. Okay. Alan, would you like to? There's a reply from Bruce. Agree that a table of technologies. Yeah, I think
what they're asking that is really how many mRNA you can detect. Like, what is the sensitivity of the technique? Like, how many copies do you need to be able to detect something with your technologies?
Yeah, right. I mean, there are different a range of different spatial cellular resolved spatial methods now, and they do have slightly different sensitivity. And I mean, the first question is how much of the RNA is retained in the tissue. That's sort of that is partially a tissue quality question. How much is still preserved at the time of analysis, and how much is lost during tissue preparation. And then and of the RNA molecules that are present, so what's the fraction of those you can detect? And I think there are really, I mean, methods, and I think also including our method, we could probably detect them all. So that's and clearly the single molecule fish based method seems to be very close to that, detect all the RNA molecule that in preserved in the cells. So that can be really high. I think that another challenge there is maybe partially unsolved, I would say, is how to assign these molecules to cells. To see what is the boundaries of the cells where these RNA molecules resides. In some tissues, cells are really very crowded, and it's not that easy to get the stainings that would clearly tell cells apart. And then, of course, it may be difficult actually to with certainty locate an RNA molecule to the right cell. That particularly may be difficult in axons and dendrites of neurons, which are really thin and may be difficult to track. So I think that's maybe a more fundamentally difficult thing. But I think with the 3D imaging, I think that could partially be, I think that would be really useful to be able to track the molecules better.
Okay. And for the protein, maybe Ali, you can say something, but I think no one has combined for now like spatial transcriptomics together with immunostaining, for instance, and say, okay, if there's only a few copies, can I detect a signal with my antibodies? So I don't think this has been done. So in terms of being able to tell how many proteins there are in a single cell, I don't think it has been done. So I don't know how we can tell what is the sensitivity of the technique. Maybe you can answer, I don't know.
I agree. I mean, it's hard to say. I mean, especially it comes to clearing and looking at large tissue, we are away from seeing single mRNA or protein. So we are more at the cellular levels. There should be some abundant proteins and labeling. Otherwise, it will be difficult at this very large scale. I mean, we are talking about a tissue of 10 centimeters. It's very different than a section that is being imaged by high resolution confocal. So at our scale, actually, we don't really have ways to image few copies of mRNAs or proteins.
Thank you. So I think the next popular question is directed to Max, actually, which is basically getting your thoughts on the advantages and disadvantages of spatial profiling technologies like, for example, slide-seq, compared to imaging technologies like in situ sequencing for single-cell resolution.
Yeah, and maybe also for you, Joachim, could fill in there. So slide-seq and has the it covers the whole transcriptome, so it's a completely unbiased approach. You would sample the whole transcriptome in spatially resolved way, and the spatial resolution is sort of dictated by the spatial features that you use to probe the tissue. So it's very scalable. It typically doesn't go as deep into the transcriptome as the in situ based methods. The slightly worse sensitivity. So that's, I guess, and also of course it has spatially worse resolution. It isn't submicron resolution. I think that would be really difficult with that approach. And I don't know, Joachim, if you would like to add something there.
Sure. I could just add to the first question that you read. I think there's you could separate the technologies into the ones that look at the RNA directly, and they have a higher sensitivity as compared to the methods that use the cDNA step. By using the reverse transcription, you lose sensitivity. So that's a game changer a little bit in terms of sensitivity. If we talk about slide-seq or spatial transcriptomics, I fully agree with Matt's comments. I mean, these are exploratory tools to look for the landscape for things. And obviously, there are reports that you could come down to sub-micron resolution with those type of features on array scale. So I think that will be a potential use of that type of technology going forward, but it's not really a usable technology today. But therefore, the technologies that capture cells and groups of cells, I think the tools are very nice for exploratory things, but with the limitation then that you don't really have the same sensitivity as focusing on a panel of genes.
Thank you very much. So it sounds like the different technologies are all complementary and provide different resolutions. Yeah. So it depends on the biological question. So I want to go back to the original question that was directed to Mats, but I think also probably relevant for everyone on the panel with regards to spatial and non-spatial cell-cell interaction. I mean, looking at the thread, this is basically asking with regards to comparing algorithms that are available or databases that are available for receptor-ligand interaction, such as CellPhoneDB or NicheNet, and how those can be applied when you actually have spatial information as well in this form of technologies and platforms.
Yeah, I mean, so I think the spatial data could just maybe strengthen these predictions about the interactions of cells, because you could really see neighboring cells that might be, really in contact with each other, and if they do have really have expression of the receptor and ligand, then that it's still not the real evidence that they are actually interacting, but at least they have the capacity to do that through these that mechanism, and also for maybe more. But here I actually think that it would be powerful to add on detecting the ligands as such on the protein level, the real chemical compound that is signaling. So I would love to see that kind of data integration on top of the RNA expression profiles. both in cells in suspension and in tissues.
I could add on a little bit to that. I think there's enormous potential in the marriage between the single cell data, which is very crisp, and the spatial data by slide-seq or spatial transcriptomics or anything like that, because you could actually map down with the probability that certain cell types are present in a spatial context. Again, as Mats says, that's not a proof, but it actually gives an indication of that these are spatially localized, and if you have multiple observations of the same, it becomes much more solid, I would guess. But a full creation of interactions would be proteins, obviously.
Thank you. I just wondered if Shao wanted to add anything to this question with regards to understanding cell-cell interaction in spatial sort of imaging approaches.
Yeah, I think cell-cell interaction is definitely a very important direction for the spatial data. I think the current limitations is first, as Matt just mentioned, so it's sometimes it's hard to visualize cell-cell boundary, which means even we see there is a receptor and a ligand expressed in neighboring cells, sometimes we still cannot confidently draw the conclusion that this kind of like a ligand receptor interaction is actually happening without visualize the actual protein-protein interactions. So I think, yeah, now with all this like data integration, we can make predictions about what the potential interactions are. But eventually, I think it relies on like a protein-based detection of those actual protein-protein interactions to confidently draw the conclusion this is happening. So I think it will need further experimental method development. And another issue is for some of the tissues, we can deduce cell-cell interaction from their spatial distance in many other organs. But in the brain, cell-cell interactions are not necessarily happen if they are just next to each other. They have many like long-range interactions mediated by projection, etc. And in order to visualize those cell-cell interactions, again, it's very important to visualize the cell morphology, cell boundary, and also those like cell surface proteins.
That's brilliant. Thank you very much. I'm going to go very quickly now to Ali because there's a specific question for you, which says, what tissue clearing technique works best with standard immunostaining methods?
There are, of course, a lot of different tissue clearing technologies, different approaches. We use the ones with organic solvents, and actually they have been quite successful to do large deep tissue immunolabeling. The version that is called iDisco, using 3Disco plus deep tissue permeabilization and antibody labeling. Most of the time it should work well. There have been different publications in mouse embryos to mouse organs and to human embryos by Alain. So that would be our first one to go, but there are definitely other ones. If it is a particular one doesn't work, one can go and check other clearing technologies, because clearing chemistry is different. Based on that, epitopes of the antibodies might change differently. Sometimes an antibody of interest may not work in a particular clearing, so one may need to check. But in our hands with disco clearing, we were able to actually make most of the ones that we were interested in working.
Yeah. If I can just add something, what is interesting with the solvent-based clearing is that you do the immuno before you do any kind of treatment. So you don't really touch the antigens, unlike other clearing technique. So basically, it seems to work with antibodies against basically any types of proteins, because the clearing is done afterward. So that's why our experience and what Ali is doing as well suggests that that's really a nice technique, because you kind of don't touch anything before you do the clearing. You just do your regular immuno and then you clear.
Sure. I mean, actually, I meant here the chemistry with permeabilization, because now we have this based technology where actually we need the permeabilization, of course, before the clearing. But you're right, overall the organic solvents come at the end in our case.
So thank you very much. There's another question which is a general question, and it's with regards to the availability of the single-cell RNA sequencing data sets. The question says, are they uploaded quickly on the site, or do we need to request access? And just to sort of iterate that there are a few major sources of this data. The Human Cell Atlas project is building a data portal that will ideally have all the public normal human tissue available. And that's the website on the answer, which is data.humancellatlas.org. And there are other resources that you can look up, which is where some of the kind of like contributing groups have uploaded their data sets to, including repositories such as GEO, Array Express, and the Broad Institute. But perhaps I could ask this question now, not just for single-cell RNA sequencing data set, but for the imaging data sets that are also available, and see what the answers from the panel will be for that. So maybe Alan or Ali, with regards to the large imaging data sets.
So we actually, there's an important point, at least for structural imaging, we have been sharing the original data that we have as much as possible. The problem with the imaging data in particular, it can go in the order of terabytes easily. It could be like 50, 100 terabytes data of one paper. It is not, actually, I don't know any platform that I can share such data. So what we are doing is we are putting some of the examples, not 50 of them, but 3, 4 of them, and say you can request more if you want. For example, we have the whole brain the mouse brain vasculature as an atlas, which you can go and download at least 3, 6 of them, and if you need more, we can provide. But actually, this isn't a problem to be solved. Maybe Alan can also comment on that. It is for, as a researcher, for us very hard to get access to such platforms that we can share 50 terabytes like easy.
Yeah, so I fully agree. I mean, it's really a problem for what we do and what everybody's doing in terms of even live imaging. And so there's a discussion that will occur at some point next year or at the end of the year. How can we compress the data? Because it's really getting too big and we might not need to get them as raw data. So I know that Ali is working on compression algorithm, and that will be probably a solution, because for mapping cells, we don't need to get like all the information. It depends on the resolution. It's really getting problematic. And if you want to do tissue clearing yourself and 3D imaging, it's something to consider when you're going to buy a system. You're going to need a lot of servers, storage space, etc. So it's going to be very costly. But it's really a very important problem that we're facing now, and we're working on solutions and to try to make it easier, sharing images using like some online viewer and cloud system. But this has a cost.
Thank you. Sharon, did you want to say something?
Yeah, so I think regarding data sharing, because we also encountered this problem, and what I found is most of the people, they are only interested in the processed data, which is the single cell gene spatial matrix plus some 3D coordinates for each gene, not necessarily the raw imaging data. And even we share with them the raw imaging data, they may not have the workstation or the right like computational resource to reprocessing them. So I think if we can all reach some standard about imaging pre-processing, then we can definitely make the processed file more available to other people, and only share the large raw data when the request group has enough computational resource to process those.
Brilliant, thank you very much. Now a question for all the panel. Actually, this is a good one to debate. How do you make a reference benchmark for human tissue? How much variability, reproducibility, biological and technical reproducibility have you seen in mouse tissues to get an idea of what might be expected in human tissues? Is it all qualitative, or are there quantitative aspects that need to be addressed to make the data reproducible? I'll open it to everyone on the panel.
I guess there's two aspects, structural and then molecular transcriptomics, proteomics. Probably structurally, one could expect a little bit more consistency because 3D structure of organs similar, and then vasculature. Of course, they will differ, but there would be more, probably like one could potentially describe, for example, a kidney using mathematical formula, the vascular tiny capillaries around glomeruli per distance and so on. This would be relatively easier, but I guess the transcriptomics part would be more difficult. I guess this expert should answer that part.
Well, I agree, and maybe particularly because of the sheer size of at least adult tissues. By necessity, these samples of tissues, and then it's almost inconceivable that we managed to take out exactly the corresponding piece. I mean, it's almost philosophically hard. While mice, for example, we can do whole mice or organs easily. Then it's easier to then, but once you have your data, you can sort of align and compare. I think much easier. But development, maybe the organs are of a size that we can sort of do comprehensive maps, and then we will see. We can much more easily align and compare between samples.
Yeah, that's what I wanted to say. It's clearly for development, it's going to be easier because, I mean, if you know the Carnegie stages, for 8 weeks, basically all embryos, they kind of look the same and develop exactly along the same way. So it has been quantified for decades now. So normally the chance we can really do the whole organ and tell you how many cells were in specific organ at a specific time with hours of precision. So I think that we should be able to answer this quite soon, and it will be probably much easier than in the adult. So for the development perspective, that should be possible to have a reference and have an idea about viability also between embryos, if there is one.
And I guess that also brings an additional complication in the sense that with development, you have the different gestational stages. And so reproducibility then is not just about one organ at one time point, but also the temporal aspects, which will be a challenge, but certainly a worthwhile challenge for us all to pursue. I want to move, sorry, was there another comment?
Yeah, just a minor comment. I think we're still just seeing the tip of the iceberg in terms of molecular heterogeneity. So I think the first years of spatial benchmarking will tell us much more how we should organize, how we should record, and how we should organize the framework for doing spatial work, actually.
Cheryl, do you want to say anything about the reproducibility element from your experience?
Yeah, so my experience is, I think there are two levels. First, it's like bioreplicates within one technology. I think that's easy. You just need to quantify those parameters like cell types and gene expression profiles. And I think if we see consistent results within biological samples using the same method, we can have the first level of confidence. And the second level is the inter-method, cross-method validation. That certainly is harder, but it's also necessary. So we cannot say one method is ground truth, the other is biased, but I think each method may have its own bias. But as long as we see the reproducible results cross method, there will be a higher probability that may be something true. So I think developing computational algorithms to enable cross-method validation will be very important, and that algorithm should address like, okay, assume we both have bias, then how do we discover the ground truth across multiple methods. So, yeah. But I have to say I have very limited experience with the human tissues. I'm only handling mouse tissues, so I guess my view may be limited.
Thank you. So I'm going to move to the next question and try to merge 3 questions into one. And this all relates to basically the tissue clearing protocol, or kind of like tissue processing protocol. The first is, does the tissue clearing protocol significantly degrade RNA? And then second one is, does it affect the preservation of the different cell types? And then we've also got a comment from Bruce about how these kind of like processing methods affect the extracellular matrix structures and how this information can be preserved or lost during the analysis. So I'm open it to the panelists, with the first aspect being RNA degradation from tissue clearing.
Alan?
I thought you would answer. No, we know it doesn't, it depends again on the tissue clearing technique you do. So there are techniques based on clarity where they've been showing that you can really do like clearing an mRNA in situ in 3D, there's no problem. We know now that ionoscope can be combined with tissue clearing. And we also know that at least for the type of technology that Ali developed, you can clear a tissue and still do in situs behind it. But it's something we are working on. But yeah, there's a chance that the mRNA is still present. I don't know if some of it is gone or not, but it's compatible. And there are published papers where they've done it.
Yeah. So definitely there is some decrease. I mean, at least if we talk about organic solvent-based clearing, some decrease in RNA quality, then it depends what kind of questions to be answered. And in situ hybridization seems to be actually working relatively not bad after organic solvent-based clearing. Related to cellular structure after clearing, in general, tissue clearing methods, they change somehow the volume of the tissue. Either they enlarge or they get smaller. Rarely they are fully unchanged, because it goes through different processes, from sometimes in dehydration to sometimes embedding into water solutions with sugars that expands. So, but overall, what we see, I guess others as well, the change in the volume is ratiometric. For example, if you think about the brain and the cortex expand the same way with the hippocampus, it's a little bit like expansion microscopy. If we use organic solvent, it shrinks more or less ratiometrically. So what was the third one?
So the third one was the cell type. So there was the RNA, and then the extracellular matrix, and the cell types.
ECM, we didn't really work on it so much after tissue clearing, I must say.
But we know that if you do, we do staining with isolating before, we do also staining against collagens, and it works very nicely. So it means that the antibody you still use, or the staining that you do against a lectin, it's still there. So yeah. It means even if some of the matrix is gone, if there's still some left, we can still do a very nice IB4 or anti-collagen staining. So it's not completely gone. You remove lipids, but anything that is bound to protein, I mean, seems to be kind of okay. So it's not a normal matrix, but it's not completely dissolved.
True. So there is the fixation, of course, cross-linking with PFA, usually that fixes a lot of things, and organic solvent-based clearing, they do remove lots of lipids. If you're not particularly interested in lipid structure like myelin, most of the ECM proteins and everything should be actually in their place.
That sounds like the tissue clearing doesn't detrimentally affect ECM or the cell types. So that's good. Then we're moving to the next question, which is now going back to, well, actually it covers everything. So the question is, which are the best practices when integrating in situ hybridization, immunofluorescence, and spatial transcriptomics? Any offer from our panel members? Joachim?
Yeah, so as Mats alluded in his presentation, we have this Human Developmental Cell Atlas project ongoing on brain, heart, and lung. And then we're actually collaborating a number of groups with different disciplines. So we try to plan these exercises using we try to section the same organ at the same time for multiple technologies. And by that way, we could develop a best practice to link the different data sets from the same organ. So a lot of the technical noise will be avoided by doing this. So it's our experience that works pretty nice, actually, because then we could take the exploratory methods to find patterns, and then we could use in situ sequencing to confirm those patterns, and then we could add on the protein on the same organ. And that's very, very sexy, I would say, because you combine all the different omics in the same material. And that's easily done, actually.
Mats and Xiao, would you like to add to this discussion point?
Yeah, I basically agree that if you are planning to apply multiple methods to the same sample and try to integrate them, the best way is you prepare the sample at the same time, and I think the integration results will be better than you apply different methods to different animals or human tissues and then try to come back. So I think the best practice is to plan the experiments very carefully to make sure you can collect the sample at the highest quality for each of the those measure modalities. We have tried to do like in situ sequencing and in situ immunostaining at the same time. And some of them are successful because the quality of the antibody is good. Sometimes it's not so successful. So I think it also requires some like a testing experimental run, just like to make sure you validate each reagent, especially antibody, because that's the most variable part you're working with, and make sure every part is good before you put in a whole experiment with precious samples. Yeah, and also regarding the quality, I think typically if you are working with in situ hybridization without reverse transcription, it's typically okay. You fix the sample and store for a few weeks or months. But with reverse transcription based method or our method, even we don't require reverse transcription, but we do require enzymatic access. The fresher the sample is, the better the results are. So I think fresh tissue samples always works better than old samples. Yeah.
Thank you. So the next one is very important for everyone in the panel. And this is the question says, spatial analysis is considered as a challenging algorithmic field. What is for you the way to build the right research team? So who wants to take this on first in terms of how to build a team to undertake spatial analysis?
It's a tough question. Of course, I guess in general, in large data, algorithms, the best methods to analyze data is not necessarily there, but is constantly being improved. I guess with the recent advances in machine learning, we have a solid ground, I would say, at least in terms of structural analysis. Clearly, deep learning-based algorithms outperforms anything. So therefore, there I would definitely bet everything on machine learning. For transcriptomics-based analysis, I guess that's also now coming more and more.
Okay.
Yeah.
Any other offers? Yeah, Xiao.
Yeah, I can. I think because I just started my lab 1 year ago, I can tell you my struggles. It's definitely hard, especially if you have been like wet lab works, and then suddenly you do have a computational challenge, and if you don't solve it, you don't have your experimental results at all. So I think regarding building the team, first there are two possibilities. First, you you can recruit a postdoc student who already have the computational experience, and then you can learn together. There are not so many, but some good publications come out from the field. You can download their codes from GitHub. That's a starting point. And you can request their raw data. So you can practice, reproduce their results to make sure you do the computational steps correctly. The other is through collaboration. Like, for example, I'm in chemistry, there will be less exposure in the field in computation. So sometimes it's hard to recruit the right person in your own lab. Then you can always set out to like engineering school or a computational biology field, try to see if you can establish any collaboration to help you regarding the data analysis. But yes, that's actually a hard part. But once you have it, it can stably being used for a while. So yeah, I think that's the essential part. But yeah, that's just my own experience.
And any input from the experience of the SciLife Lab? Matt?
Yeah, well, yeah. So I mean, my lab is quite experimental, and I've just actually sort of started establishing a more computational skills, and my strategy has been to recruit biologists skilled in computational methods or computational people that are skilled in biology, and they have to sort of be a little bit of generalists and mainly actually work as contact persons with really computational scientists. We're really working on a collaborative mode. So we're typically very open sharing data we generate with anyone that is interested in trying to develop better tools, because there are certainly still early days, I think, in the computational methods dealing with spatial data. So that's well, my approach is to try to help others developing the tools we all need.
Yeah, I could just add one more thing. I think what I've learned, I mean, we tried to develop technology and we tried to also develop the methods, but as long as we don't provide data, it's hard to attract computational expertise. The day that we have data lying around and we don't know what to do with it, then we're very attractive, both for recruitment but also for collaboration. So data is driving the development, I would say, to a large degree.
So no, that sounds excellent. A multidisciplinary approach and kind of reach is probably something that anyone who wants to go into the field has to think about. I'd like to move to the next question, which although this question is specific with regards to disco and whole mouse imaging, and asking and saying that the whole human and mouse brain atlas provides a nice reference framework for a coordinate system to map cell level information to an organ. Are there others for other organs? Should we be thinking about a standardized coordinate system? And that's from Trevor McKee. And this is something that's come up as a common coordinate framework within the discussions of the Human Cell Atlas. And it's a good one to sort of like get the panel to give their thoughts on this. Ali, would you like to go first with regards to, since your name is mentioned?
So I think that's a very good point. We have very nice detailed subdivisional maps of human brain, very 1,200 parts defined in Allen Brain Atlas, but we don't have that much details for any other organ. So now we are having cellular level information access to the organs such as human heart. I think we will come to this point. Combined with especially some molecular information, structural information, there will be more detailed maps and coordinates of human organs that I would see that coming. So, and I think we should aim that from probably developmental to the adulthood, there will be defined maps of the other human organs.
Yeah, and if I just add something, because there's also some ongoing efforts to try to first do like imaging using other technology, like even including like fMRI or the kind of non-invasive techniques. And then if you get the samples, to try to correlate the image you got before clearing with the clearing. So maybe you can then try to also match both. Like, so for the brain, for instance, if people have like callosum, corpus callosum defect, and you do imaging of those people, then can you correlate it with 3D image? So maybe it would be then very interesting to try to then use what you learned from the 3D imaging clearing to put it back to the clinic to better interpret what you see on your images. So that's really something that will be very valuable as well.
Great. And I think there's also efforts on this front to kind of integrate the suspension single-cell RNA sequencing data to the spatial data sets using the coordinates that have been defined at the macro level as well. So I'm going to move on to the next question, which is specifically directed to Mats regarding the probabilistic method to assign the cell type. Is that a reflection of the uncertainty of cell type due to lack of signal or differentiation potential of a cell?
So, yeah, all right. So I think the way we have implemented the probabilistic cell mapping is really on mature, fully differentiated cells in mice. That is what we sort of developed the method for. And then I think you can sort of, then you can make the match between the molecular information you extract from every cell in your tissue, in your image, with the average expression of a cell cluster of this mature differentiated cell type. In development, there will be a lot of these intermediate states and then transitions and cells on the move. And then I think that one can't really use or apply the PCI-SEC, the probabilistic method in the way we did it in the mouse. And one have to sort of map it to a trajectory somehow and define these gradients of expression in a lineage, and then try to pinpoint where on this trajectory this cell most likely is. But it will be, but the probabilistic aspect of it is really that we're matching the molecular data we record in situ with the molecular data that has been recorded with a more extensive molecular profiling method like single-cell RNA sequencing.
So perhaps
the uncertainty you have in the single-cell RNA sequencing data will sort of be forwarded to the spatial interpretation of that data.
Yeah. So in addition to kind of like using the suspension dataset to derive forward the annotation of the in situ sequencing method, maybe Joachim might be able to also elaborate on where in the sort of spatial transcriptomics technologies where you may not necessarily always have just one cell within the capture area for how you would assign cell type.
Sure, I think I alluded to that a little bit earlier, that you could actually take advantage of the crisp cell types defined by single-cell sequencing, and then you could, by probabilistic or other methods, call which cell types are present within one spot of a spatial array. And that's very, has been working extremely well actually in our hands. And there's a lot of activity of trying to merge single-cell data with the spatial data. I just want to point to one additional advantage, is that we could actually take advantage of the information in the image as well. So what we are trying to do is also learn from the H&E image that we have, what kind of structures correlates with transcriptional patterns. And by doing deep learning of the images, we could actually also get another feel for the cell types by using that type of information on top of everything else. And that's, I think, extremely promising what we could do, the different data sets to increase the precision in cell types prediction.
Okay. Xiao, do you have anything to add to the question on assigning cell type based on the different methodologies?
Yeah, we also tried some efforts using StarMap, and so one key parameter we try to get a sense of is how many genes we need to map spatially in order to successfully integrate our spatial cell typing with a single cell typing. And it definitely depends on the tissue type, but the basic conclusion is the more genes, the better. And so I think with 10 or 20, it's okay. But it only when the cell type are super clear in that tissue. And with 100, it's typically good. With 1,000, I think it's better, but there is some marginal benefits in addition to 100. So I think just for the purpose of the cell typing, if for all targeted spatial mapping methods, my own experiences, I think somewhere between 100 and 1,000 will be a good number, and below 100 is also great, but the confidence may be lower.
Brilliant. Thank you very much to the panel members for answering that question that came from Eripan Salvador Martinez. So I'm conscious that we've only got a few more minutes left. There's one comment to actually say which will integrate into the resource area of the for this session, which is from Jason Swedlow, who says imaging datasets of this size can be published, viewable, downloadable, and integrated on the image data resource, open microscopy website, and the bioimage archive. Both are run by from EMBL EBI. And then very finally, two questions that's going to be merged into one, which is basically how long does the imaging take, and how long is the fixation process? And then specifically for Ali, would it be possible to use Chanel histology for antibody labeling of intact mouse brain tissue?
That's a good point. One can use the Chanel method also to study mouse organs. We didn't do it so much because there is already existing protocols for that working very well, like iDisco. And related to imaging time, I'm not really sure what exactly the sample was asked. It could be for a mouse brain, it could be a few hours. For whole mouse body now, it was the initial one that we published 2016, it was 1 month. For one mouse, now we are down to 12 hours. So there is really good scaling there. And for whole human kidney, it took us also, I guess, about a week or something to image. So while, of course, it takes time, but it's also accelerating well, combining with microscopy system like Mesospin, where you can really scan things much faster at high resolution. So I think we are in a reasonable time in general in a few days.
Brilliant. I think this is all the time that we have for questions. And we have a few slides to finish with. Firstly, thank you all very much for joining us for the Human Cell Atlas Developmental Spatial Cell Mapping Seminar. Special thanks to the program committee. Quite a number of us are on the committee, and the HCA Executive Office, particularly Christine Schrank, Tracy Andrew, and also Scott Sasson, who's basically been the man behind the audio visuals today. So thank you very much. Many thanks to our speakers, Mats Nilsson and Ali Ertug, and our panelists, together with the speakers, Arlen Shadotal, Joachim Lundeberg, Xiao Wang, and thank you very much for the wonderful talk and the panel discussion. There will be a follow-up Asia and Australia seminar, and that will be on August the 3rd, 8 to 9 a.m. British summertime, 4 to 5:30 p.m. Japanese Standard Time. And the purpose of this is to allow our collaborators in Asia and Australia to engage with today's speakers. And there will also be an extra short seminar and with additional guests, Hiroki Ueda and Linda Richards. We hope you can join that. And the feedback survey that we asked earlier for the active poll, please fill that out if you can. That will be really helpful for us. Today's recording will be posted on YouTube and also Bilibili, and the registration for the Australia follow-up will go live on our website very soon. So look out for it. And thanks once again, everyone, and for our speakers. We'd also like to announce the next HCA developmental seminar, which will be on stem cell, organoid models, and regenerative biology. We have fantastic guest speakers, Barbara Trotland and Carl Kohler, and this will be sometime in mid-August. Stay tuned for date, time, and the panel members. For more information on this seminar series, if you've enjoyed it, and if you want to see the recording of today's seminar, please go to the website humancellatlas.org/HCA meetings. And please join the HCA project registry to find out more about what we're doing. Again, the website is there. Thank you very much, everyone, for joining us today. And if you have any suggestions and offers on how we can do better, any ideas, we want to know. Do take our feedback survey at the Slido website. If you go to polls and select HCA Developmental Spatial Cell Mapping Feedback Survey. Thanks very much.